Oh, that lab report's not that long. Okay. Well, I can't help you there. Uh-huh. All right. Let's see.
Will we be outside next lab?
Hopefully, that's the plan. Um, I don't really like sending students out so the first week. So here at the beginning of the semester, we have some flexibility in kind of what we do. So we could do the bean lab at the beginning like we did this semester, but had it been warmer those first couple of weeks, we would have gone outside and done observations and then done the bean lab later. Because, I could say, oh, suck it up and be tough and be out there in like 18-degree weather with snow on the ground. But, the fact of the matter is, I don't want to be out in 18-degree weather with snow on the ground, and I would have to be out there with you. And, it's not a competition like who's the toughest field biologist. So,
It's not me.
we'll look at the weather, and hopefully weather won't be so bad that it prevents us from going out and doing the work. As we get further along in this semester, we will get to the point where we won't have to worry about weather. It'll just be nice. We'll do our field work. We'll come back. We'll analyze our data, and everything will be hunky-dory fine. Um, but until then, we'll play it by ear. I'll look at the forecast
It says snow on
at the
Wednesday.
big. What?
It says snow on Wednesday right now.
Oh, of course. So that's another issue. We have to drive to Smithville Lake because that's where the most geese populations are in this area.
Where is that
It's north of here and west. You go up on I-435 and then go north on 169. The bad thing about Smithville is kind of the further away you get from 435, the less likely the roads are to be plowed. And the other thing is, I would rather not take you out in the field and have us end up in a ditch. That would not be good. So I'll look at how weather is shaping up at the beginning of the week. I'll probably give you some inkling as to what we're going to do in lab on Thursday, on Monday. But then if conditions change between Monday and Thursday, we'll just be flexible about revising that. So yeah, that sounds like a big non-answer, which is kind of what it is. But the goal is to be in the field next week. Um, oh, I'm not projecting, am I? PLC 110. Why is PLC 110 not one of my options? Oh, there it is. All right. Okay. Um, any questions on what we've done up to this point? Clearly, Tinbergen's 4 questions are of importance. So knowing what those are, knowing what they mean, and I was going, so let me back up. Every Thursday morning before lab, I meet with Emily Sawyer because she has a course conflict, a scheduling conflict with the course. So she's watching the videos, and then we meet once a week to kind of talk through anything that she has difficulty with. As we talk through this, and I told her this at the beginning of the semester, I said, I'll ask you, what clarifying questions you want to ask me, but what I'm not going to do during that time is I'm going to say, you know, how's everything going? Do you understand everything? Do you have any questions? And then if you don't have any questions, then we're just done for the day, because that's probably not going to be very productive for you, using this time to make sure that you're staying up on things. So what I did this week with her yesterday morning is I asked her if she had any clarifying questions, and then I proceeded to ask her things like, well, what are Tinbergen's 4 questions and what do they mean? And then she was struggling with that a little bit. I'm like, okay, don't think of them and kind of trying to define them. Think of them in the context of a real example, like birdsong. which is the example that's in your textbook. And once she did that, it was much easier for her to think about what each of those questions meant. So you need to know what Tinbergen's questions are. You need to know what they mean. But on an exam, you might find it actually easiest to answer what those questions mean by actually thinking back to a concrete example that we talked about in class to illustrate what they mean. So as you study this stuff, relating it to concrete examples a lot of times will help you understand it better. The other thing that we're doing here at the beginning is we're revisiting some of the core concepts that every biologist who walks around should kind of know, just as part of their walking around knowledge as a trained biologist at the level of having a bachelor's degree in biology. The theory of evolution via natural selection and knowing how it works and what it is, who derived it, all that stuff, that's certainly something that you guys just as walking around biologists should know. But also knowing something about the four forces of evolutionary change, which is part of what we're going to talk about today, is the kind of thing that just every biologist walking around who calls himself a biologist should know. And if somebody said, in what ways can organisms, populations of organisms change over time, you should be able to trot out the four forces of evolutionary change like that. Boom, 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 boom. In the same way, Central Dogma is one of those things that just every qualified biologist should know the basic way in which we go from genes to phenotype. You don't have to know all the nitty-gritty details, but you should certainly have the broad picture. So a lot of the things that we're talking about here at the beginning of the semester in animal behavior are things that you've seen in other classes. I know that you've seen in other classes because we covered it in classes where I had you as students. And also I know that you've had Central Dogma when you took genetics, because I know what Dr. Reynolds teaches in genetics, because I've taught genetics with her in the past. So, so some of this is review, remembering things that you learned before and have forgotten, that have fallen by the wayside, and then this repeated exposure to them slowly over time gets them stuck in your head in a more permanent sort of way. So we talked briefly about the theory of evolution via natural selection. Selection is one of the four forces of evolutionary change, but there are others as well. So what are they? I just gave you the first one, selection. So selection is one of them. What's another one? Okay, so migration. It's important that you're making the distinction between those things because people get this confused all the time. If you have two locations, location A and B, you live in location A. Immigration with an I is individuals coming from another population into your population. Immigration with an E is individuals leaving your population and going to another population. So this is IMM migration. This is EM migration. So different words for different directions of flow. But the direction of flow is always referenced to you. I'm in this population. Who's immigrating in? Who's immigrating with an E out? Other forms of evolutionary change. Mutation. What are some things that we need to know about mutation to think about it in an evolutionary sense? Is this a process that leads to quick evolutionary change? No, it's something that doesn't happen very often, and as a result, this is relatively slow. Same thing with migration. Organisms move around all the time, but each time an organism moves, it's only carrying its set of genes with it. And so mass migrations can result in lots of evolutionary change, but a lot of times the level of migration, that once again is a relatively slow form of evolutionary change. What are we missing? Genetic drift. What are some things that we need to know about genetic drift? Part of what we need to know is in the name itself. It's random, it's not directed. Changes that are directed would fall under the category of selection. So this is change that moves in random directions rather than being directed in a particular way. Under what conditions is random genetic drift more prominent in terms of affecting evolutionary change? When do we expect to see random genetic drift affecting the evolutionary trajectory of a population? Under what conditions of that population do we expect it to be acting? Does population size have anything to do with this? In large populations, what happens to genetic drift? Exactly. In large populations, there's so many individuals that little changes due to just random death don't really have much of an effect. But in a small population, one or two individuals dying randomly could have a potentially large effect on the distribution of alleles in that population. So this is something that is of more concern in small populations. So let's go through and explore these things. So there they all are: mutation, genetic drift, migration, and selection. So before we delve into those, I want to back up a little bit and just review what the central dogma is, because all of these things are essentially things that affect the representation of genes that occur in a population. You have a gene. You guys know the tongue rolling gene? Tongue rolling. Masks off for just a sec. She can't do it. Now, we won't make fun of Avery for not being able to roll her tongue, but what this tells us about Avery is it tells us that Avery is a homozygous recessive for the tongue rolling gene. So if we have a gene, and we'll just give it the letter T, the dominant form of this gene, the dominant allele, is the allele that allows you to roll your tongue. Makes you a tongue roller. The recessive allele, the recessive version of this gene, prevents you from doing this. Now, as a result, I could be a homozygous dominant genotype, or I could be a heterozygous genotype. Both of these will result in tongue rolling. If you are homozygous recessive, though, you are a non-tongue roller. So that's the gene. There's a gene. The gene has two forms. Those forms are called alleles. So let's get all the terminology up here. Alleles, genes. What's a good definition for a gene? Good walking around definition for a gene. Once again, I know for a fact that you've been given this in other classes, so take a stab at it. What's that? Okay, so it is DNA, but it's a sequence of nucleotides in DNA. Okay, what do they do? What does a sequence of nucleotides accomplish? Okay, so sequence of nucleotides in DNA that code for a particular protein. This is the definition that I learned in high school. Why is this not the greatest definition in the world? It's not a terrible definition, but it's not as good as it could be. Does DNA only code for proteins? What else does it code for? Well, so a protein is just a sequence of amino acids. It codes for RNA, so it codes for things like ribosomal